Hi, my name is Katie and this is my dog Caysie. Today I wanted to discuss computational biology, and I am going to show you how it can be used to develop new medicines to treat disease. Computational biology is related to bioinformatics, which is the use of computers, software, and mathematical models to process and integrate biological information. Computational biology is the process of analyzing and interpreting biological data. Bioinformatics and computational biology are two relatively new fields that grew out of the Human Genome Project. This project was begun in 1990 and ended in 2003 as an effort to sequence the entire human genome. A flood of information was produced from this project, and it spawned these two new fields. Genes are comprised of DNA, which is a polymer made up of monomers called nucleotides. DNA has two sugar phosphate backbones and nitrogenous bases held together by hydrogen bonds to form a double helix. One of the main functions of DNA is the synthesis of proteins. DNA transcribes mRNA, which leaves the nucleus, and works with ribosomes and tRNA in a process called translation to form a polypeptide, which is a protein. Proteins are polymers made up of monomers called amino acids. There are 20 amino acids which comprise all proteins. Each amino acid has a different side chain and different characteristics. Some are hydrophobic, some are hydrophilic, some are acidic, and some are basic. The primary structure of a protein is the amino acid sequence that makes up the protein. Then the protein will fold into its secondary structure, which could be either an alpha helix or a beta pleated sheet. The secondary structure is formed by hydrogen bonds between the repeating constituents of the amino acids. For example, the red oxygen here bonds with the white hydrogen here. Then the protein will fold into its tertiary structure, which is formed by hydrogen bonds between the different R groups of the amino acids. Sometimes two proteins will join together to form a quaternary structure. If there is a mutation in the DNA, it could result in amino acid substitution, resulting in an amino acid with a different side chain. This would disrupt the protein's structure and hence its function. Proteins are the main agents of biological function. Therefore, it follows that proteins should be the key targets of molecular-based studies when trying to understand biological disease states. The disease pathogen I'm going to focus on is the HIV-1 virus. This is the virus which causes AIDS. In general, a virus is comprised of DNA or RNA surrounded by a protein coat. In the case of HIV, we have a retrovirus. Shown here are the envelope proteins. These bind to the host cell. Shown here are the RNA strands. And here's the reverse transcriptase, which transcribes a viral RNA template into DNA. Through the years, proteins and their interactions were determined experimentally. Example methods include X-ray crystallography and yeast-2 hybrid. With bioinformatics, scientists started entering the results from these experiments into databases which are now available to everyone. Some of these databases include PubMed, a database of biomedical literature, GenBank, a genetic sequence database, Intact from the European Bioinformatics Institute, UniProt, a catalog of protein sequence and function, and DrugBank, a drug and drug target database. These are some of the databases I used in creating this protein-to-protein interaction network. It shows a complex set of interactions between HIV-1 proteins and human proteins. I used network analysis techniques, which utilize metrics such as these, to analyze my protein-to-protein interaction network. Some of these metrics include degree, the number of links a node has, betweenness, the number of times a node acts as a bridge between other nodes, closeness, clustering, and characteristic path length. By studying these interactions, we can identify disease pathways in key areas to direct pharmacological research. Highly connected hub proteins are more important to virus survival than a protein with a small number of interactions. The nodes with the highest degree are shown in this chart. Note, E and V at 135. Let's look at the viral envelope protein GP160. It interacts with many human proteins, including CD4, which is the primary receptor protein, and HLA-B, which presents foreign antigens to the immune system. so large hub proteins may be good targets to obstruct virus function. I searched the drug bank database and found 4 experimental compounds which target the GP160 protein. GP160 is ENV. 3-aminoalanine, beta-3-serine, beta-3-cysteine, and 2,4-diaminobutyric acid. In conclusion, my project outlines a process for using computational biology to guide pharmacological research. Consult genome databases to find proteins from species of interest. Consult interaction databases to construct protein-to-protein interaction maps. Use network analysis techniques to determine key nodes in proteins. And use drug and chemical databases to find candidate compounds for pharmacological research that affect those proteins. I use the HIV-1 virus as a given pathogen. However, this is a general process that can be used for any given disease. Caysie, isn't it fantastic that I was able to develop a process that can be used for any disease?